welcome everyone. I'm going to flip over to my slides now. So Fanny's given you a wonderful exposition of the utility of intraoperative OCT, and indeed it really is in many respects quite transformative, particularly in respect to gene therapy delivery. So my history of involvement of using OCT actually started with Bioptogen, the handheld OCT, which I've had probably for the last 7 years. And we were using this pretty extensively for examining children, young children, where you might be able to have a look at for foveal development, for example, if you were interested in evaluating whether a child might have albinism or something like that. And in fact, in a number of clinical trials, it's had a huge amount of use. And it's a very useful device for a mobile patient. It's allowed us to start looking at craniofacial patients, the synostosis patients, and try to see whether or not we can correlate their optic nerve appearances on OCT with the intracranial pressures. And so with that in mind, when the on-focus OCT came along, I was very keen to have it, and I've been a keen Leica user for many years anyway. So the surgical equipment that I use intraoperatively, as we've said, I use the Preveo 8 with the on-focus intraoperative OCT. I too use the Biom 5 with the EVA Nexus from Dork. I'm currently using the 2338 subretinal polytip cannula and microdose system from Med1. So I'm going to tell you a little bit about a 3-year-old child. I tend to do most of the children under 10 in the UK. And so my experience of gene therapies is different to those of Fanny in the sense that she treats a lot of patients with much more advanced disease, much thinner retina, whereas I am lucky enough to treat younger patients whose dystrophy is far less extensive, and their retina is therefore much more healthy. And she presents, this 3-year-old girl, with the classic features of RPE65 disease. So she's got light staring, and their children are profoundly night blind. And so these are sort of characteristic features that you look for in children with early onset retinal dystrophies. And so light staring behavior and profound night blindness are very characteristic. She's a compound heterozygous, 2 different mutations in the RPE65 gene. And she has the classic nystagmus, low illumination visual acuity, and reduced photopic acuity. And that means her vision is 1.2 logMAR, so that's just worse than 660. She's myopic, as many of these children, many of these adults are, and she's got an absent ERG, and her pattern visual evoked potential is also significantly reduced. As Fanny indicated, we have a very similar setup. We both use dilute triamcinolone to visualize the gel, and the aim is to create a single bleb with a single retinotomy, putting 300 microliters under the retina and underneath the macula as much as possible. So we're aiming to go arcade to arcade through the fovea and ideally not touching the disc. So here she is. This is Optos Silverstone images. You can see probably some mild vascular attenuation, but as you can tell, she's got a pretty blonde looking fundus, which is quite typical for these Caucasian RPE65 patients. And these Silverstone images, you can see an intact ellipsoid there going pretty much the whole way out. So a very normal looking fundus, really. So this is her first eye, her right eye, just to show you what surgery looks like. And it's the first question that most people ask me is that how is inducing a PVD in these younger patients? And in truth, because they're dystrophic, a PVD induction has generally not been terribly difficult. I mean, one struggles a bit sometimes, and using triamcinolone, and as Fanny indicated, it can be very helpful. And here I go just inside the arcade. You can see bleb induction is very straightforward. You go for a very low, slow infusion, and that's what you're looking for, arcade to arcade. It's quite a big, quite a large bleb. It looks like it takes quite a long time to get it all in there. And then, as we've said previously, we do a fluid air exchange, and that removes any refluxed vector that's come through your retinotomy, and allows, if you're trying to manipulate your bleb, it allows you to move it more posteriorly with the air bubble. So, having seen what surgery should look like, I want to show you what her fellow eye looks like, because I think this really illustrates for me the utility of the OCT. And I'll sort of pause on that thought, because we got our on focus about halfway through the gene therapy program, having started using voretigene in I think it was 2019, the approvals came through. And at the time, I was using the Bioptogen, and I had a attachment to my microscope, and I would put it on the side, and it was a bit of a Heath Robinson setup, and I would use it very much just to check at the end. I'd move the microscope to one side, and that would place the OCT, the handheld OCT, over the top of the eye, and then you take some scans just to check placement. And so it really wasn't the same experience as what I've got now, where you can flick the OCT on and visualize in real time what's going on with bleb placement. So let's go to this video, and you can see why I think it's really important. So again, PVD induction, much as previously demonstrated, no great surprises here. I like to use the flat lens. I like having a bit more magnification. And again, you can see, so retinotomy, you're looking for a little bit of a choroidal blanching. And then I start to infuse. And the difference I'm suddenly noticing here is that the bleb is not propagating in quite the same way as it would normally do. It normally is a smooth, gentle enlargement, and yet here I'm not getting it. And I look across at the OCT, and you can start to see this ellipsoid that has peeled itself away from the inner retina and is attached still to the RPE. And like many of us who've had complications or difficulties during surgery, you're not quite sure what you're seeing some of the time. You're thinking, is this real? What am I actually seeing? And I'm actually demonstrating for you here a bacillary detachment, which is photoreceptor detachment. And I make a decision then to go and make a second retinotomy of the inferior arcade. I still have the same objective. I still want to take that bleb through the fovea. And what I'm going to try and do is extend it out now through the fovea inferiorly and make it join up with the superior smaller bleb. And I have no problem stopping, looking at the bleb, making sure I often have an assistant that helps me do the imaging. And so they're on the screen. It's a lovely touch screen, and I can visualize the bleb, stop, infuse a little bit more until I'm comfortable that I know I've got the right amount subretinally. And then I will go to air, and you'll see those two blebs slightly join up on the OCT, and that means that I've got a nice bleb that goes the entire way through the macula. And like Fanny, I will go and inspect the fovea to make sure I haven't got any foveal inversion. But in children, that's really not a problem. Macular hold is not something that I worry about in children because they have such healthy retina. And so, one of the questions that people ask is, why has this happened? And so bacillary detachment, I think, probably occurs in gene therapy simply because fluid follows the path of least resistance. And in children, where those tight photoreceptor outer segment RPE interdigitations, those are really still quite strong. This retina is not dystrophic to the degree which you would find in adulthood. And so those adhesions are so strong that actually the fluid is finding its way through the outer retina more easily than it is cleaving its way through the RPE photoreceptor space. And although this is a very unusual finding, it's not something that I've ever observed before doing this procedure. I think what you can tell is that having that OCT allowed us to make this diagnosis in real time, to change our surgical plan and to move on and place the bleb elsewhere and ultimately have a successful operation. So how has she ended up? Well, like many of the children, the youngest ones, we are trying to intervene in the period, or the critical period of visual development. So it's my strong feeling that the younger we go, up to a point at least, the more inroads we can make into their deprivational amblyopia. And this is, this is not a view that necessarily everyone shares, and there are clearly risks, surgical risks, the younger you go. But I have seen, much more dramatic improvements in visual acuity in the younger patients. And so this girl is now 2 months out. She's already improved by 2 to 3 lines of visual acuity. And I expect that she will probably continue to improve a bit more from here. And her low illumination vision is markedly improved. She's got reduced nystagmus, and she's much more confident in her mobility at all light levels. And her parents, were, needless to say, delighted. So what are my feelings about the benefits? Well, I'm a great fan of Leica optics. I think they're wonderful. I love the recording system, as many of you who are Leica users will appreciate. The recording system is very intuitive. It's very straightforward. I like the fact that I don't have to fiddle around. I just plug my USB in, I press record, and it's there. The quality of the imaging is definitely better than the previous Bioptogen engine. It's definitely received an upgrade, and it's noticeable. And it's very much easier using the automatic focus and retinal location. You really don't. It's a single push button. It finds the retina, and the quality is good very quickly. So I find it very, very useful for correct bleb placement and for checking foveal contour, as Fanny indicated. And so I'll just follow up by giving you a little bit more feedback. That her mum was very happy to share some of these things that she has noticed since having this treatment. She said she would always bump into things, the corner of the table, cupboard doors, etc. But this isn't true anymore. When climbing stairs, she always used to feel her way up the stairs with the side of her foot and along the wall. And now she just walks straight up the stairs without checking at all. And more recently, she's noticed birds on the top of a building and saw a rainbow for the first time last week. And, she will venture into rooms with no lights on and regularly proclaims, "My magic is working. I can see with no light." And you can see her here scooting along. This is something that she was not doing pre-treatment. Gene therapy, we're at the dawn. I always say this to my patients and my colleagues. We're at the dawn of an era. This is the first real demonstration of the efficacy of gene therapy, and I have no doubt that we still have a long way to go. But using equipment like the intraoperative OCT and some of these other technological developments that Fanny has alluded to earlier, I think we are making some really exciting strides using these technologies, and I have no doubt that we'll establish their utility further with greater experience. So I think with that, I'm going to move along. Thanks very much.